You are listening to the Two Disabled Dudes podcast. I'm Kyle Bryant.
And I'm Sean Bompsark. Together, we're navigating life with disability. All right, thanks for tuning in. This is the Two Disabled Dudes podcast. I'm Sean Bompsark. Somewhere on the other end of the line is Kyle Bryant, and you are listening to episode 35. Recently, Kyle and I had an incredible opportunity to visit Reata Pharmaceuticals. They're developing a drug for Friedreich's ataxia, and they're also doing a bunch of other stuff in the rare disease space. So, last month we were hanging out in Dallas area with these guys, and we had an incredible opportunity to record 2 interviews on site and, of course, meet a lot of their staff. We're super excited about the work they're doing in the rare disease space, and specifically even with Friedreich's ataxia. So we're thrilled to bring you 2 episodes from their office.
So we have a 2-part series for you. First of all, today we'll be introducing you to Reata's founder and CEO, Warren Huff, and Reata's Chief Medical Officer, Dr. Colin Meyer. In our next episode, number 36, we've got Reata's Manager of Patient Advocacy, Kara Eichelkraut. Both episodes will shed some light on the value of a strong patient community in the drug development process. So, first up, here is our conversation with Warren and Colin. We hope you enjoy. All right, everybody, welcome back to the Two Disabled Dudes podcast. We are live today with a special episode from Reata Pharmaceuticals in Irving, Texas, right outside of Dallas. Sean, what's going on?
No, it's freezing cold here.
Yeah.
Like freezing.
Yeah, or even less than
was
freezing
hot all the time, but apparently I was wrong.
It was just today.
Just today.
So the voice that you just heard was Warren Huff. He's the CEO at Reata Pharmaceuticals, and we also have Dr. Colin Meyer, who is the Chief Medical Officer at Reata Pharmaceuticals. And these guys are here to join us for a conversation today. Welcome to the show.
Oh, great. Thanks for having us.
Definitely.
Of course.
Thanks for coming.
Yeah.
But before you guys get into it, we also want to make sure our listeners know we're in a room full of Reata associates. So where are you guys at? Yeah, there it is.
Yeah.
All right.
We got a rowdy crowd here today. This is good.
It is
Awesome.
Texas.
Yeah, it is Texas. Don't mess, man. That's what I'm talking about. All right, so we wanted to talk about who is Reata, and Warren, because you're the guy who started Reata. We wanted to talk about how the heck do you start a pharmaceutical company? And so maybe if you could take us back to the beginning, like where did Reata start?
Okay. Well, maybe I'll give a little just quick background on myself, which would kind of tell how we got here. I was a lawyer for many years and, was reasonably successful at it, but absolutely hated what I was doing. I hit kind of my mid-30s and I was like, I really can't see myself practicing corporate law for the rest of my life. And I had a tech practice, so I represented a bunch of IT companies and a small number of pretty interesting small biotechs. And I found myself spending a ton of time on these small biotechs, which frankly didn't bring me much business. I was just really fascinated with what they did, and in particular the science. So to make a long story short, along the way, I made a decision to leave my practice. I got recruited by a startup biotech in Boston that was a client, and I literally left my law partnership behind, picked up my family, moved across the country to Boston, and I was the CEO of a public biotech there for 5 or 6 years. And after I got there, I realized, wow, this is like the biggest mistake I have ever made.
In terms of the company
or
the weather?
It had all these issues that I didn't know about, and all these challenges. And it ended up working out pretty well. I got the company public. We completed registrational studies, drugs. We had some hiccups. Drugs ultimately got approved. And what I would say is in that experience, this company was a single technology, which came out of a single lab. There was kind of one scientific idea, and we did all this work and raised all this money and put all this organization together, and it could all fail on one idea. And so after we kind of finished that up, I ended up moving back to Dallas primarily for family reasons. And when I moved back, I got introduced by some of my early investors in that first company to a fabulous guy named Dennis Stone. Dennis passed away last year. Many of you know. He was until recently a member of our board of directors. Fabulous guy. But Dennis and I met, and he was running tech transfer at UT Southwestern. And UT Southwestern is a powerhouse medical school. It has enormous impact in molecular biology and biochemistry, and has been the basis for many important, innovations. And in almost every case, they were getting licensed out to companies that were venture-backed in San Francisco or in Boston. And you guys have been to Texas a few times now, and in Texas, we don't like that. And so...
do it all yourself.
Right, yeah. And so people wanted to see an industry get developed here. And in particular, the University of Texas system had initiatives to help Dennis and his counterpart at MD Anderson, which is also a University of Texas subdivision with a lot of medical research and innovation. And basically, they had a checkbook to seed companies, but they had virtually no experienced management teams. And so Dennis and I started working together, and he was tried multiple times to get me to take these single project companies, to take one of those on. And I just kept thinking, no, I'm never doing that again. And I gave it a lot of thought. And along the way, I kind of formulated the idea for a company that would be focused, instead of on a single technology, would be focused on the blocking and tackling of building a great innovative company. The technologies would really be used to put the organization together, to create many products, and there would be many benefits of that. First, you would mitigate the risk of technical failure, and as you guys know, there's a lot of risk of technical failure when you're trying to do truly innovative drug development. And by the way, that was the mission. In-license and develop highly promising technologies that were very early stage and very risky, but had the potential to have high impact on basically essentially untreatable or very serious diseases, basically moonshots, and aggregate a whole portfolio of those, and then basically recruit and develop a really experienced drug development team to advance those in parallel.
So the difference between like this philosophy and that of like the regular, big pharma is that a lot of companies are built around one single technology, right? I guess that's what you just said, but just to reiterate that point, like that's what's different about Reata, right?
Yeah, that's how we got started. And then also I would say we only worked on novel. So we limited ourselves to working basically on drugs that had novel mechanisms of action. And actually, in several of them, including our NRF2 activators, in many cases, the mechanism of action wasn't even known. We were really just looking for interesting, provocative biologic activity, and we assumed that we could figure it out along the way, basically how it worked and where to apply it. And so that's when we started in licensing projects, and we had a lot of things go south on us along the way, but enough to keep us going.
Yeah. So I think that's where I want to bring Dr. Meyer in, because you guys have been together since the beginning, right? And or close to it. And so I I wanted to ask maybe both you about some of those failures in the beginning that were, those moonshots that may or may not have worked out,
Yeah. Do you want to?
Sure. So one of my favorite stories is a project that we licensed from academia. There was this really interesting compound that was discovered in a sponge off the coast of Australia. And in cell-based experiments, it had really interesting activity. It was active in cells that were resistant to radiation therapy. And as you may know, radiation therapy is used to treat like half of cancers. And so clear, applicability to a large unmet need. And when we started scaling up the molecule to put it into animals, we were planning our experiments, and we were having a scientific advisory board meeting. And one of the people on our scientific advisory board, Tom Sudoff, who was formerly at Southwestern and is now the head of neuroscience at Stanford, and actually won the Nobel Prize a few years ago, he looked at the molecule, the mechanism of action, and said, I just want you to be aware that that target in the cell is very important in neurosynaptic transmission, and you may see neurotoxicity when you put it in animals. And we said,
well, what does that mean?
Sure, Tom, great. And so we made enough of it, and I remember the day that we first put it into animals, and this was, 10 or 12 years ago, I got this call from the lab, and we gave like a single dose of 100 milligrams, per kilogram of the animal, and the animals immediately died. And
so it's hearing a story like that reminds me as a patient why I need to read the fine print.
Yeah.
And reminds me of just how thankful we are of the process.
Yeah.
Of development. Yeah. Thankfully, it was an animal, and your first test wasn't a human.
I think that's a good point, right? Because we talk about the FDA, in our minds, they're like this adversarial body, right? That's preventing us from getting to what we want. But they put in place this process where that drug is not going to be neurotoxic to Sean or Kyle or anyone else. It's, so... Yeah.
Yeah, I think it's hard to appreciate how good drug development is in terms of like assessing safety and getting molecules that are active and figuring out how they work now. There's just been a huge amount of innovation. And I think it's also hard until you've been through a few of these of like, again, how much failure there is early, right? A lot of things get tried and they just don't work out. And you guys see it at the clinical stage. But man, years of work and risk has been taken before they ever get into humans.
Yeah. So you guys have had a few failures. How do you pick up and move on after a big failure? And maybe something like that, like these moonshots, right? Part of the philosophy is that some of this stuff is going to fail, and that's just the way it goes, right?
Yeah, it was built in.
But then there's things that work to a certain point, right? And maybe they get through a phase 2 or something like that. And how have you been in that situation where something failed in a little bit of a later stage? And how do you pick up and move on after that?
Yeah, so we've actually had a phase 3 trial fail a little over 5 years ago. We were conducting work in patients with very advanced kidney disease, and so diabetics who basically are within a couple months or a few years of needing dialysis, and those patients are very sick and fragile, have a lot of cardiovascular disease, and we enrolled this trial across 300 clinical sites, 2,200 patients, and the data we had generated previously demonstrated that the drug has these very provocative, large increases in kidney function. Most people thought that once you lost kidney function, you could never regain it, and so there's a lot of interest in the community, and it was one of the most prominent programs in chronic kidney disease. And after enrolling all patients, the independent data monitoring committee that's responsible for assessing safety on a fully independent basis detected a safety signal. And I was the one who got the call when they recommended terminating the trial. And it hit us by complete surprise because we did not have a safety signal in prior trials that treated about 500 people. And so in that setting, that was our lead program. And we weren't sure if the company was going to be viable, at that time, in part because we didn't really know what had happened. And then moving forward, we basically had to close down the trial. We tried to figure out what had happened in a 1-week time period with some external additional reviewers, and we couldn't. And after many months of closing down the trial and analyzing the data, we came to appreciate that the initial safety concern wasn't actually what had happened. And so there was a concern that the drug may have been affecting the heart. It turns out the drug was promoting fluid retention. And we identified a subset of patients who were at risk for developing this fluid retention. And so through literally 3 or 4 years of work, we were able to determine that it really wasn't as bad as we thought. We shouldn't have stopped the trial if we would have known. We were able to identify patients who are at risk, and we now exclude them from future trials in our kidney disease program. And it's easy to monitor, for fluid retention. So kind of all that being said, it was a major setback. We didn't know if the company was viable. And in this setting, fortunately, it didn't. It result in us, going away permanently. And we've actually been able to revive the kidney program in part because we can isolate that risk and still use the drug, safely in other patients.
Nice. I like it.
I think that speaks a lot to the learning curve that failure presents. Mm-hmm. You can look at it in any walk of life. People always talk about, it's like you mentioned, what you get at the trial level is everything's working. What we see in celebrity or author or any other walk of life, all you see is the shots that Michael Jordan can nail, mm-hmm. not what went into practicing and the failed attempts leading up to that game-changing moment.
Yep.
So I think what you're talking about is it's a very expensive failure, I'm sure, and very time-consuming and very discouraging. But now you know what to look for, and now you know what to exclude, and it just helps move the needle forward one little bit, even though it wasn't maybe the answer we were hoping for or the end target you were hoping for, but at least it helps you understand how to navigate tomorrow.
Yeah, and what I'd say is that this is just what comes with the territory when you are trying to do really innovative development for really difficult diseases. Because let's face it, the drugs are powerful. You're modulating, new genes, trying to tackle something really difficult, and this is just the process. You have setbacks, but the promise of that drug is. It's the only drug, I believe, really ever to demonstrate clinically major reversal, essentially, of chronic kidney disease. And, that has the potential to affect millions and millions of people over time.
A different train of thought, and we'll shift gears here in a minute. But we talked about a couple of failed mechanisms. And, of course, we want to talk about some things that are looking great and promising. But before we get into any specific, what would you guys say as the founder and somebody that's been around from just maybe a few months after that or whatever it was, what are some of the most, or maybe one or two of the most exciting moments for Reata?
Well, there actually have been so many. On the positive side, I think that when we identified and in-licensed the project that led to both the renal drug and our FA drug, I think that was a pretty amazing time. It's maybe a little hard for people to appreciate, but these drugs just had amazing biologic activity, from the beginning, and we in-licensed them. And they just had these very powerful effects that just kind of jumped off the page, and nobody knew how they worked. And I think a huge thing was when we got our initial insights into how they were really working, what was really happening, how They were, affecting this target NRF2 that helps regulate your bioenergetics and your antioxidant status and your inflammatory status, and how kind of fundamental the biology was. For me, that was a big moment. I think when we discovered the initial findings in CKD, was a big moment. And frankly, I would say our phase 2 data in FA, where we actually saw frank recovery, a neurologic function in patients on drug for only 12 weeks. I shed some tears that day. I mean, I think a lot of people around here did. It was a huge moment. So, and there's been a lot of things. We went public a year and a half ago, and I think that was a big moment for us because it was after the failure of that large phase 3 years ago. So we fully recovered, worked it out, took the company public, and that was a nice from a corporate development side. Was a really important moment.
Thank you, Warren, for sharing those. Colin, is there one that sticks out differently to you, or would you say those are all same major points?
I'd say similar points. Some of them are, of course, are the same, but also I think the successes we have in our clinical trials. That's really important for us, for the patients and the communities that we're working in. And so I think every time we have a positive trial, it's just further validation that we may be able to help, not just those people in the trial, but potentially thousands or tens of thousands or literally millions of patients. And it's not just that we're helping them now. As once a drug is approved, it's available for all patients and basically for eternity. And so you can make a huge impact on humanity by developing new drugs. And so it's always really exciting when we have a success in one of our trials.
Sure. Well, part of the reason why we're at Reata today is obviously our listeners know Kyle and I live with the Friedreich's ataxia, and you guys have been a partner in research for FA. But before we get into the specifics of the drug that you mentioned that you guys are developing and talking about in the trial, Colin, tell us a little bit about you. How did you come to this company? What's a little bit of your background?
Sure. So when I was growing up, I was interested in medicine. My uncle was an oncologist, and so I always thought that I wanted to go to medical school. I went to this math and science school to basically finish up high school, went to college early doing that, and I thought I wanted to do an MD PhD program and maybe be like a physician scientist. After I finished that, I went to University of Virginia to finish my undergraduate degrees, worked in the lab for a couple years while doing that, and thought that that was the most valuable experience that I had while in undergrad, but I really didn't want to work in a lab, and so I said, okay, I'll focus on the MD side of MD PhD. Went to medical school here at UT Southwestern in Dallas, and really enjoyed learning about medicine, but thought I would get bored practicing, basically doing the same thing over and over again, and developed an interest in the kind of business side of healthcare and medicine. And I was networking, and I went to a conference at the Federal Reserve here in Dallas about the economics of biotechnology, and I met Dennis Stone, who Warren mentioned what he was working with to put the company together. And this was probably a couple years before Warren started working with Dennis, and Dennis started mentoring me through medical school. And I decided to go to business school, overlapping medical school, and then made the decision to do something different than practice. And so Dennis introduced me to Warren, and at the time, Warren was looking for someone with a kind of general background who he could hire cheaply, and so that was me.
Minimum wage.
I see. All right, that makes sense now.
And so that was a little over 15 years ago. Joined, and so I've been working together since then.
Nice.
Awesome.
Someday we'll have to get you above minimum wage.
Yeah. So are you still a PhD flight? You don't have to answer that.
Well, Sean, so based on your car ride yesterday, I think
something tells me he's investing well.
Yeah,
he's made some good decisions along the way.
Yes. So real quick, can you tell us about your car ride yesterday?
Yeah. So Kyle Colin here is an incredible driver, and you left out your hobbies, but we'll get to that in a minute.
Sure.
So there was, some scheduling, whatever, changes. So Kara actually picked me up from the airport, brought me to the office, and then Colin was kind enough to drive me to the hotel. The hotel, apparently, from what I understand, is like an hour and a half away, but it took us 20 minutes to get there. Okay, it's really not that far, but it did take about 22 minutes because I got to ride in a Dodge Viper. Colin owns a Viper, and I've loved those cars since I was a kid. A neighbor had one, beautiful, always saw it, never got to ride in it. I got to touch it and like look inside the window, but never got to ride in one. So and they're not made anymore. So and I just think they're the prettiest looking car. So when I found out Colin had one, I begged him for it. Right.
Sounds like fun, and we're glad you're here and alive.
There were no police or anything. We were,
it was fine.
No one was injured.
No one was injured.
No property destroyed.
No, I'm a very safe driver. I'm trained.
We rolled the windows down. It was kind of like a natural facelift.
Nice. So, Colin, you drive. Your hobby. One of your hobbies is to drive cars, right? I mean, you drive on the track and stuff. Can you tell us about that? Like, how does that work? Right? Like, that's not a normal thing that people do, is it?
It's illegal for him to drive on the street now.
It's the only place I can drive, and so no. I've always had an interest in cars. My dad always had kind of interesting project cars, and so I think I inherited that liking of cars, but not the ability to work on them. And I went to this track day a few years ago and was able to drive on a closed circuit, and it was amazing. It's something that I never realized that you could do, and it's very, of course, different than driving on the street. A lot of people, look at sports cars and they say, well, how could you ever actually use them on the street? And you really can't. And so I found a car, this GT3, which is basically a street legal race car that I go to these closed circuits, and it's in the Porsche Club. And so it's very structured, and it's more like a gentleman's race. It's not technically racing. You're not going wheel to wheel. And so there's all sorts of rules because you don't want to destroy your own car. But just the amount of effort that it takes, you can't focus on anything else, and so there's no way for you to think about anything else because if you did, you'd crash your car.
Sure.
And just the kind of G-forces and excitement of it, and the camaraderie with other people who do it. It's a really fun hobby.
Can there be camaraderie if I just have a poster of a GT? Like, I don't have one, but can I join the club?
How about you come next time?
So, have you ever crashed? Like, does that happen?
Oh, yeah, it happens. But it usually happens either when people don't know how to drive, or they don't know the abilities of their car, or they don't know the track. And so the tracks I go to usually don't really have walls, and so there's a few places where you could hurt your car. Of course, I've gone off the track, at 115 miles per hour in a spin, but into grass, and so this didn't hurt me or the car. And if you're going to be able to drive quickly, you need to know how to get to the limit and stay at the limit. And in order to do that, you kind of need to know where the limit is, and you don't know where the limit is until you get beyond the limit.
Wow. It's like, that sounds like Top Gun stuff here, man, right?
It's like a whole nother episode. So before we get into RT408, Kyle, tell us about your car ride.
I rode in a Jeep Compass, which is a small SUV, super, super nice.
Awesome.
And we drove the speed limit, and it was really great.
It took you, an hour and a half to get there? We were waiting for a really long time. It
should have taken you 10 minutes. No.
We didn't gain any time on the Google estimate when we...
I thought you always gain time.
That's just how it works, right? All right. So, one of the things that we're really interested in for obvious reasons is RTA 408, and it's a drug that is designed to battle Friedreich's ataxia, and it's in clinical trial right now. and lots of our friends, and I have participated in our moving the drug along, and it got some pretty good results in the part one of the phase. Anyway, let's hear. Where are we at with RTA 408? And actually, you know what? Before that, where did it come from? Like, how do you discover a drug? Like,
yeah, actually, so that was one of the moments I mentioned earlier when we in-licensed this project. This whole class of drugs was invented by a famous scientist at Dartmouth named Dr. Mike Sporn. And Sporn spent his career discovering kind of how your tissue transforms into cancer from injury. Basically, the tissue, the molecular pathways involved in tissue response to injury. And he started this kind of remarkable project years ago where he took basically molecules from natural sources that had weak anti-inflammatory and anti-cancer properties, and he worked with a chemist, and they did this kind of beautiful random walk, what we call medicinal chemistry campaign, where they make structural modifications to the molecules, and then they kind of test to see what the effect of those modifications are. And they used this assay or test. Basically, they activated one of the most important classes of your white blood cells that control inflammation called macrophages. They activated them like they were being invaded by bacteria, and they looked at the effect of the drugs to essentially suppress or resolve that activation. And they did a whole lot of work without much activity, and then they made this one change to one side of the molecule. They put this, chemists all know what's called a Michael acceptor on the A ring of the molecule, and they got this 10,000-fold increase in potency in this assay. And so they were like super potent at resolving inflammation. And that's where the original discovery came. And then we came along, we in-licensed the project. Some additional academic labs, Dr. Sporn's lab, our internal people, our research group are all working on how do these work, and our medicinal chemists looked at the structures and thought that they could be improved, particularly for neuromuscular diseases, because the original molecules that we in-licensed did not cross the blood-brain barrier very well. And so our medicinal chemistry team invented 408 to solve that problem, to have the same pharmacology as the earlier molecules, but to be particularly appropriate for neuromuscular diseases. And that's where it came from.
Okay. And so can you describe briefly why does RTA 408 work on FA?
Yeah, so after we had in-licensed the molecules, we and our collaborators in academia identified the target, NRF2. And our drug, is an NRF2 activator. It's the most potent set of drugs that do that. And then the NRF2 literature was just blossoming with work suggesting activity and importance of NRF2 in multiple different settings. And there are now 4 different academic labs. I think most of them are funded by FARA, most if not all, that have demonstrated that in FA, NRF2 is suppressed. And as you know, the mutation of frataxin results in mitochondrial dysfunction, too much iron in the mitochondria. The mitochondria shift from producing energy to basically to turning on inflammatory pathways. And our drug induces NRF2, which should be activated, but somewhat paradoxically, it's suppressed. And so a few years ago, there were some additional insights from some researchers at University College London. And so Andrei Abramov, who's funded by FARA, as well as Paolo Giunti, who's a physician and sees the largest cohort of FA patients in Europe. And they demonstrated that NRF2 directly controls mitochondrial function. And so we knew that the drugs could affect inflammation, but there was this central link made to, energy production in the mitochondria. And then Paula, so Dr. Junchi, then harvested some cells, took biopsies from FA patients, and then gave them to Dr. Abramov, and he demonstrated that RTA408, or omavaloxolone, or OMAV for short now, sorry to be confusing, basically is able to restore mitochondrial function in biopsy samples from FA patients. And so we were working with them, Farah and Dr. Lynch were working with other investigators who had similar insights. And so we then started working with Dr. Lynch and Farah to design the first part of the MOXIE trial, which is a phase 2 program that has 2 parts. And as you know, we reported data last June. Dr. Lynch gave the presentation for the first part of the trial, and then and we were able to demonstrate in that trial that the drug did in fact induce NRF2 in patients, given RTA408 or OMAV, had biological activity, and we saw improvement, as Warren mentioned earlier, in neurological function according to the modified FARS score. On the basis of all that data, we made a few more modifications to the protocol, the second part, sent it to FDA, and they now have agreed that is a registrational trial, meaning the trial that we're currently enrolling will support approval, likely in the U.S. and other territories, if we're able to hopefully replicate what we saw in the first part of the trial and demonstrate, clinical activity as well as appropriate safety. And so I think the bottom line is, that trial is actively enrolling, and there's a lot of enthusiasm, and we really appreciate the help of you guys and FAIR and the patient community, because once we enroll the trial, we have to wait 48 weeks until all patients finish, and then if the data are positive, we immediately go to regulators to try to get the drug approved, so there could be a first approved drug for FA.
So we're gonna talk to Kara in a little bit about patient engagement specifically, but I wanted to ask you guys if because I'm hoping that part of the patient community is listening right now, right? And so was there a specific thing that you wanted to let the community know about what's going on, or whatever you want to the patient community? What would you say?
I would say that if it weren't for the patient community, no drugs would ever be developed. And so, we have been, really appreciative, of FARA because they've done an excellent job. And we work with many different patient advocacy groups, and FARA is the most helpful, most effective group that we've ever worked with, to organize the community, to educate the patient community about research, including clinical research. And while we can design the trials, we really need obviously patients to participate. And if they don't, we'll never, or no one will ever have an answer if their drug potentially could be an effective treatment. And so our message to the community is that it's extremely important that they're aware of what clinical research is and means, and specifically for us, if they want to participate in our trial, that would be awesome. And so it's extremely important because we need to get an answer, and the patient community needs treatments. And so the only way to do that is to enroll our trials.
We'll have all kinds of information in our show notes to help people find out how they can enroll if they're interested.
Yeah, we can put links to recruitment notices and all the different sites that are available that people can go to and numbers to call and stuff like that. So, yeah.
And I think maybe one last thing to mention about that is that in trials that support approval, they're placebo-controlled, meaning patients will get inactive placebo. And it's really unfortunate that has to happen, but it's necessary in order to evaluate whether or not the drug works and is safe. And so in our trial, half of patients, so of the 100 patients we need, half will be on placebo. But if patients participate in the trial and they complete the trial, they get access to the drug. And so if they happen to get randomized to the placebo arm, they will get access to the active drug. Then obviously the faster that we enroll the trial, if it's successful, then the faster the whole patient community can have access to the drug. And so, Alex, I think FAIR's done a great job educating patients. There's some aspects of clinical trials like placebo or frequent visits, to the investigating sites is not fun, but it's a necessary element.
Maybe completely off topic, but I will encourage those folks out there that are interested in clinical trial. I actually participated in part one or phase one or
Part one.
whatever you call them. Incredible. I use the UCLA site for most of my FA related stuff. They're great to work with, and then of course knowing that RTA, Reata was behind that, they were phenomenal to work with too. Not that you actually interact a whole lot with the drug maker or the manufacturer, but an easy trial to be a part of, and actually really exciting, I think, to see a little bit of how that world works. So good job, guys.
Thanks. We appreciate your help.
Yeah,
And in the trial, because I participated in the same part that Sean did, you get to ride a recumbent bike, like a station, and I love that. That's my favorite part of the whole trial. One thing about the bike though is that some people don't have the ability in their legs anymore with FA. That's just an unfortunate thing about the disease, right? And the progression of the disease. And, I think inclusion and exclusion criteria they're a tough thing, but to sort of deal with, right? But talk about the importance of inclusion and exclusion criteria in a clinical trial and why those are there.
Yeah, I think it's a really important topic. And what most people need to appreciate is that when you're doing a clinical trial, it's an experiment. And so everything has to be tightly controlled. And the last thing that we want is to get a false negative, meaning the drug probably worked, but the trial wasn't successful and it doesn't support approval. And so even though we hope that our drug could work for all types of FA patients across the whole spectrum, age range, disease severity, we have to have a really homogeneous set of patients in the trial. And so everyone does this, and we've been particularly strict in our trial. And so you have to be able to ride the exercise bike, and like Kyle said, some patients can't. You have to have a modified FARS score that's within 4.5 points, and some patients won't. You have to be between a certain age range, and some will fall outside of that. And you even have to go off some of the off-label vitamins, antioxidants that you're on. And I know that that's hard for some people, but what we've learned from a lot of the past trials in FA, unfortunately, is many have not been successful, is that there was a lot of noise and a lot of variability. And so we want to make sure that we have a really consistent set of patients so that if there's even any signal, that we can detect it and then statistically or mathematically make the case that it is real and therefore would support that the drug is efficacious. And so if you're unsure if you would qualify, I encourage you to reach out to one of the sites. If you're not near one of the clinical sites, we pay for your travel, and so pick one that you want to go visit to and have vacation a few times.
Is there one in Fiji?
Well, we were talking about Bora Bora maybe, but not yet. And so I think it's important that people are aware of this, and if they don't qualify, it doesn't mean that that the drug may not work for them. It's just we want to be really careful and do everything we can do to maximize the chance of success, with this trial.
Yeah, good deal. Yeah, that's what we all want, and that's the thing I love about the relationship with Reata and the FA community and Farah, and is that everyone's rowing in the same direction. We're all in this for the same goal, and we're gonna get there together, no matter what, and cross that finish line together. And there's gonna be one heck of a party, right, Warren?
there is absolutely Fiji.
All right. Well, of course, Colin, Warren, thank you so much for hanging out with us today and for allowing us to come hang out with you and telling us a little bit about your story, of course, your company and the drugs that you're working on. We appreciate what you guys are doing, and we're just honored to be a part of the journey for a bit.
I'm glad we're doing this together.
Yep, and thanks for helping.
Yeah.
Of course. All right. Well,
And we appreciate all of you, all of Reata, So thank you so much for being here, for having us, I guess, right?
So another live episode of the Two Disabled Dudes podcast. We'll see you next time. All right. We hope you enjoyed hearing from both those incredible guys. We definitely had a great time chatting with them. Coming up in just a couple weeks, Kyle and I will be attending Rare Disease Week with a handful of friends. This Rare Disease Week takes place in Washington, D.C. on Sunday, February 25th. The week kicks off with the screening of The Ataxian. This screening will be followed by a discussion and a bit of a Q&A between Kyle and I, the executive producer, Tom Hamilton, and Fair's research director, Dr. Angel Martin.
We hope some of you can join us if you're in the area of Washington, D.C. To register and find out more information. You can find links in our show notes for this episode. We'll be recording an episode while we're in DC during that Q&A session. So look for an episode on that coming up soon. That should be fun.
Sweet. Well, until then, we'll talk at you later.
Thank you for listening to the Two Disabled Dudes podcast. Find us online at twodisableddudes.com. And please subscribe and review on Apple Podcasts.
We'd like to hear from you. You can find us on Facebook, Twitter, and Instagram.
Special thanks to our audio producer, Jake Tompkins, who also composed the music.
Until next time, keep living with urgency.